Good afternoon and good evening, everybody. My name is Ian Coates. I'm your facilitator there for Biology 1022. Welcome to week 4 blood. Everything is looking good. You're going to see a little bit of repeat there from last week, and which tends to be the case as we move on through the course, because these organ systems, they don't act independently. Okay, they act with other organ systems. So you're going to see in here, there's going to be some endocrine, again in this, and throughout every single PowerPoint there throughout the course. So yeah, we're just having fun. I hope you guys are doing all okay. If you let me know if there's any issues, you guys have my email. And as always, I just go straight through, but please feel free to stop this at any given point in time, watch the videos, read the book, all these kinds of things, all in conjunction with this PowerPoint. And to no further ado, let's get on with blood. This is a fun one. This one here is a springboard to most other organ systems, so let's go. Blood contents. Blood and types of connective tissue. Okay, it is a type of connective tissue, exactly kind of the same thing as bone. Okay, they come from the same area. Okay, and you're going to see there that blood is actually made in the bones. We talked about that there week 1. Blood itself is a complex mixture of cells, chemicals, and fluid. Transport substances throughout the body, helps to maintain a stable interstitial fluid environment. You'll remember that term there last week there when we introduced the immune system there. Interstitial fluid is the space in between cells. And as well, distributes heat throughout the body from the muscles, right? The blood includes RBCs, WBCs, platelets, and plasma. We'll get into all these different functions there later on. Blood volume and composition. Plasma is a mixture of water, amino acids, proteins, carbohydrates, lipids, vitamins, hormones, electrolytes, cellular waste. Yeah. Plasma is that yellow ooze there that you'll see there, like from an oozing cut kind of a thing, all right? Blood hematocrit, okay, that's the blood test there to determine how much you have, is normally around 45% RBCs with about 55% plasma, okay? Show you a slide right here. It gives you a nice picture of what this is all talking about here. And oddly enough, we only have about 5 liters of blood. That's it. So if you nick an artery in the right spot, it will not take long. That's about 2.5 Coke bottles. Okay. Yeah, it's not a lot of blood. You can bleed out pretty quick. You'll see here, this is just the routine operation there of taking blood. Okay, then you centrifuge it down, spins it, spins it, spins it. And what happens here relative to their densities there, things tend to separate. The heaviest goes to the bottom, lightest stays at the top, and naturally there at the top there's your plasma. Okay, it's just fluid there, and as we said, this is what's going on with plasma right in there. There's a lot going on. Okay, water, amino acids, proteins, sugars, fats, vitamins, hormones, salts, cellular waste. Yeah. Then you have the RBCs down here, so that's about 55%, 45% or so. And then you'll see that you have this buffy coat, and these are your WBCs, okay, and platelets as well. And just a microscope slide of what all is going on here. Perfect. Just a nice shot here of the separation there between the formed units. Here we're talking the RBCs 45% as opposed to the plasma, the non-formed units, okay. Kind of dissolved within the plasma, 55%. So now you see the formed units here. Okay, there's your platelets, RBCs, WBCs, which can then be further broken down. And most of these there you would have heard there last week, which will continue on again there this week. Neutrophils, eosinophiles, basophiles, monocytes, lymphocytes there. And there's your different percentages. And not that I would ever ask you percentages. Plasma, the constituents there of the plasma, as we've already noted. Nutrients, vitamins, hormones, there's your different gases there. Be careful of this one there if you're a scuba diver. You've heard of the bends. Proteins involved in the blood, albumins, globulins, and fibrinogen there. Fibrinogen, again, you'll hear when you're talking in conjunction there with platelets. They're involved in stopping a cut and preventing a cut and closing a cut. Erythrocytes, beautiful word there for an RBC, interchangeable, know it for sure. These are bioconcave discs. Okay, you can see there's kind of a hole in the middle, and that's the same on either side. It's like a donut there that's about to have its hole punched out. And these bioconcave discs that contain about 1/3 oxygen carrying hemoglobin by volume. When the oxygen combines there with hemoglobin, turns bright ruby red, oxyhemoglobin. Deoxygenated deoxyhemoglobin tends to be a little darker. The RBCs there, they don't have a nuclei, so therefore there's no mitosis. All right. The RBCs there, they can produce ATP through glycolysis alone. Since they don't have any mitochondria, there's no use for they can't use the oxygen there that they are carrying. They cannot use, yes, something's wrong here. And ignore all. There we go. Can I use that? There we go. That makes a little bit more sense. All right. Very good. Again, 5 liters there. Typical red blood counts out there for males is between about 5 and 6 million per cubic mil and millimeter. Okay, and then for females there tends to be about 4 to 5 million. Okay, the number of RBCs is a measure of how the blood's oxygen carrying capacity details, right? When we're talking about RBC production and its control, here you're talking basically the kidneys, the forefront, liver as well. Okay. In the embryo and the fetus, RBC production, erythropoiesis, great word, occurs in the yolk sac, liver, and spleen, but after birth it occurs in the red bone marrow of the bones. Okay. Erythropoiesis occurs in the bone marrow for adults. Okay. The average lifespan of an RBC is about 4 months. Tends to be about 3 to 4 months, somewhere in there. The stages of RBC formation from hemopoietic stem cells, okay, hemocytoblasts. The total number of RBCs remain relatively constant due to. Oh, hi guys, back again. There, hey, with the marvels of technology, this new program I got, I can pause this recording and take care of business. Usually, I would have to stop it altogether, but everything is great. You guys will remember there the negative feedback involved. Okay, when we're talking about blood production, you guys will remember negative feedback there from Bio 1021. The whole idea of it kind of looks like a teeter-totter, a seesaw, right? It goes back and forth. It's always monitoring 24/7, okay, where it raises the level and then it decreases the level and then it raises it up. Well, that's what's going on here. And the hormone erythropoietin, okay, short form EPO, which is released there from the kidneys and liver in response to the detection of low O2 levels, okay? And we'll show you there the negative feedback there, loop there, the next one there. Excessive increase in RBCs is called polycythemia, okay? The opposite there is anemia, okay? Low blood count, anemia, excessive RBC count, polycythemia. And you see what's going on here. Low oxygen there is detected in the blood, okay? Sends a message there to the kidneys and the liver. Kidneys for sure make erythropoietin, the hormone there, which goes into the bloodstream, which then eventually makes itself to the red bone marrow, which then says, hey, kick up RBC production, which it does. Now the body has an increased oxygen carrying capacity, which then turns this whole system off, right? Then this would whole shut down, then this would shut down, and away we go. All right? Beautiful little negative feedback. Dietary factors affecting RBC production. Vitamin B12, folic acid are needed for DNA synthesis, so they're necessary for the reproduction of all body cells. Okay. Iron, but this is mainly recycled and kept. Okay. So, but when you're talking females, though, through menstruation, they will tend to lose quite a bit of iron on a monthly basis, so they can suffer from iron deficiencies. Okay. As stated earlier, deficiency in RBC production or quantity, the word is anemia. There's a couple of different anemias out there. One of them there you might have heard of, sickle cell. Okay, talk about those there later. Destruction of RBCs. With age, anywhere between 3 to 4 months, RBCs become increasingly fragile. They become damaged because, you're passing through these very narrow capillaries. And capillaries, as if you don't know this by now, these are the only place in the body where, the blood is able to exchange, things, constituents, sugar, proteins, oxygen, carbon dioxide. This all happens at the capillary level, and it's quite narrow. And basically the width of an RBC, flowing down a capillary. And it gets bumped, it gets damaged. So eventually, macrophages, they come from the liver there and the spleen to phagocyze these damaged RBCs, Chomp, chomp, Pac-Man. Hemoglobin there from the decomposed RBC cells converted into hemoglobin, where the heme is decomposed into iron, which is stored or converted into biliverdin, bilirubin, which are excreted there in the bile. The globin is broken down into amino acids and reused. You'll see there that everything is reused. Breakdown, there's your phagocytosis, chomp chomp, broken up into the heme, into the globin, then changed into bilirubin. And then bilirubin is converted into pigments there, parts of the feces. Iron is recycled, amino acids are recycled, and the whole process again. This is just a shot there of the NRBC getting the chomp chomp here. It's getting phagocized. Just moving on to the next slide here. Kind of says the same thing here. Nothing too different. But phagocytosis here of an erythrocyte by macrophages there in the liver. Again, lifespan. Blood vessel macrophages there, breaks down these guys, breaks them all down into their constituents. Bone marrow requires B12, iron, amino acids, copper, cobalt, and extrinsic factor. And extrinsic factor is an unidentified factor, provided by the gastric mucosa. Interesting. That is necessary for absorption of B12. It's called extrinsic factor because it derives from an external source in foods and is not synthesized by the body. So, it's a little bit of information there. Hemoglobin. Hemoglobin is made up of 4 protein molecule chains connected there together. Normal adult hemoglobin contains 2 alpha globulin chains and 2 betas. 2 alphas, alpha, alpha, beta, beta. In fetus and infant, and in infants, beta chains are not common. Here they have alpha and gammas. As the infant grows, though, the gamma chains are gradually replaced by these betas. You guys can read up more on those. Myoglobin versus hemoglobin. Myoglobin we introduced there a couple weeks ago there when we were talking about muscles. That's the muscle version of hemoglobin there. It's an ability there for all these muscles to store a bit of oxygen, so that in the case of a dinosaur or a bear, we're able to get free for a certain amount of time. But that is very limited, a limited supply, and it'll get you through 100 meters. But then after 100 meters, you start going into oxygen debt, and you need to breathe harder because you need the oxygen from the atmosphere. But both are proteins there that contain iron. We know that. One is in the muscles, one's for the blood, Hemoglobin is present inside RBC cells and delivers oxygen to tissues. Myoglobin is found in skeletal and cardiac muscle only, not smooth, and supplies with O2 in stressful situations. Hemoglobin has less affinity for oxygen as compared to myoglobin. So you think about that. You breathe in oxygen into your lungs, the oxygen then jumps into your blood, And then that blood travels that blood carrying the oxygen travels all the way to your muscle. And then how does that oxygen leave the hemoglobin and jump onto the myoglobin? That's because the myoglobin has a higher affinity. Myoglobin has a greater affinity for oxygen than hemoglobin, so therefore it will jump and stay on the myoglobin rather than the hemoglobin. Hopefully that makes sense. Pause that for a second there if that didn't make sense. I'll try and let's try to say it again a different way. But myoglobin has a greater affinity for oxygen. So that's why when the hemoglobin is carrying the oxygen through the blood, the oxygen is able to jump because it's more attracted to the myoglobin than it is the hemoglobin. Okay, kind of makes sense. But the one thing there you got to be careful of, my hemoglobin, which is right here, hemoglobin. I know you guys are students, you guys have just moved, you guys maybe have new flats. I want you guys, if you guys are in the basement, if you guys are anywhere in your new flat, new house, know where your carbon monoxide detectors are and make sure that they are working, because oxygen, hemoglobin has a high affinity for oxygen. Hemoglobin loves oxygen, but you know what it loves more? Sadly enough, carbon monoxide. And if it had the choice, it would take carbon monoxide over oxygen. So you just want to be careful, okay? And make sure that your carbon monoxide detectors are working. Any issues with that, please get a hold of me ASAP, okay? Number 5, again, myoglobin has a greater affinity for O2 than hemoglobin because the latter, hemoglobin, delivers oxygen to the body. Myoglobin does not, all right? Myoglobin is kind of a terminal end for oxygen. Number 6, myoglobin consists of a single polypeptide chain, and hemoglobin consists of several. Okay. Myoglobin is high in diving animals, like seals and whales. And you think about that, right? They take one big breath and then they're down under the water for 15, 20 minutes, half an hour. Okay. Myoglobin's got to be enormous. And you'll see what's going on here. What we've got, there's your number 1, start here, 2, 3, 4, 5, 6, 7, and then back again. The small intestines absorb nutrients. Blood transports the absorbed nutrients. See, we're transporting it. RBC cells are produced in the red blood cell. Okay, here we are. In the red bone marrow, Number 4, red blood cells circulate in the bloodstream for about 120 days. That's where we're at. And then 5, macrophages phagocyze and break down the red blood cells. Here we are. Chomp, chomp, chomp. Right? And then they break down. Hepatocytes break down hemoglobin. 7, iron is recycled via the bloodstream. Biliverdin and bilirubin are secreted in the bile. You'll see the bile right here. That gives it its characteristic hue. Bile is secreted in the small intestines. There you go. That's it. That's the bit of blood recycling. Okay. White blood cells, aka leukocytes, help defend the body against disease. They're formed there by hematoblasts, okay, and hematopoietic stem cells, okay, and red bone marrow, okay. Hemocytoblasts, sorry, it was a different word there. Hormones there that stimulate white blood cell production fall into 2 categories. There's what we call interleukins and CSFs, colony stimulating factors, okay. White blood cells can leave the bloodstream to fight infection. Absolutely. You won't believe how much these things can jump. Okay, and we introduced there last week the whole idea there that they can move in between blood vessels. They can jump from one to another. Those blood vessels, they have holes in them. Lifespan tends to be around 12 hours for WBCs. Okay. There are 5 types of white blood cells that are circulating and are distinguished by size, granular appearance of the cytoplasm, shape of the nucleus, and staining characteristics. The types of white blood cells are granulocytes. These include neutrophils, eosinophils, and basophils. And then we have agranulocytes. These are monocytes and lymphocytes. Okay. Neutrophils there, they have a purple-stained fine cytoplasmic granule and a multi-lobed nucleus. Okay, they comprise about 60% there of all leukocytes. Okay, these guys here are first to respond. Eosinophils there, they have a coarser granule there that stains a deep red. A bilobed nucleus makes up about 1 to 3% of the circulating leukocytes there, okay? Basophils have fewer granules than they stain blue. They account for less than 1%. Those are your basophils there. Lymphocytes there are long-lived, have a large round nucleus, and account for about 30% of circulating leukocytes there. Monocytes are the largest of them all, okay? Have variably shaped nuclei, make up about anywhere from 3 to 10% of circulating leukocytes there. Okay. Functions of white blood cells. Now again, we're not going to go too crazy into every single one of these there's PhDs written on. We're just taking a flat rock and throwing it against flat surface, okay? Just picking up what we need and moving on, okay? The functions there of WBCs, the leukocytes can squeeze between cell linings, okay, the blood vessels, okay? Very, very important, because if there's disease on one side of the body and these guys here need to get there fast, okay, diabetes, okay, and this is the whole idea, and we'll show you this right here. You'll see how these guys, they squeeze through. Here's your blood capillary, right, and your leukocyte squeezing through. Diapetesis, all right, and they can attack the bacteria and debris there just by getting through, okay, wherever they need to get to. Neutrophils, monocytes are phagocytic, okay. With monocytes there, they become macrophage, engulfing the large particles. Both of these contain many lysosomes there that help break down organic molecules. You remember those, okay? They're able to break down, okay, foreign particles. Eosinophils there, they moderate inflammation and allergic reactions, as well as defend against parasitic infections, okay? Great. Love that slide. There's your connective tissue, and there's your leukocyte squeezing through, needing to get to where it needs to go. Continuing on there with functions of the white blood cells there, the basophils migrate to damaged tissue and release histamine, okay, to promote inflammation, and heparin there to inhibit blood clotting. Heparin, it's also basically known as rat poison, okay. They give this there to to elderly people there. It's an anticoagulant, okay. It stops the blood from coagulating, and you see it inhibits blood clotting, and it's good there if you have high blood pressure, or you're prone to clots there. You want to keep that blood flowing, right? So you want to keep it's a blood thinner. But the trouble with this is that these people are very prone to bruising, and they're very prone to, if they get a cut, a simple paper cut can be life-threatening, because they don't have any blood clotting agents there to work to stop the blood from flowing out of that tiny little cut. Lymphocytes there are the major players in specific immune reactions. B cells produce antibodies. Plasma and T cells are also produced. And there's a nice little bit of information there on heparin. All right, rat poison there. Good little gets into some deep vein thrombosis, pulmonary embolisms, arterial thromboembolisms there. Have that a read. Very, very interesting stuff. White blood cell count. Normally, a cubic millimeter of blood contains anywhere from 3.5 to 10,500 WBCs. Leukocytosis occurs after an infection when excess number of leukocytes are present. Leukopenia, too few WBCs, occurs from a variety of conditions, including AIDS. Okay, AIDS is this is the disease there that this virus, very, very insidious. It targets the WBCs, and that's where it replicates. And so by targeting these WBCs as a cell to replicate, you're basically inactivating the person's immune system. It's a dangerous one for sure. Okay. Moving on into, just continue on here with the blood cell count. A differential white blood cell count, also known as DIF, okay, can help pinpoint the nature of an illness, indicating whether it is caused by bacteria or virus. A differential white blood cell count lists the percentages of the types of leukocytes in a blood sample. So just based on their different function, whether it's going to go after bacteria or not, we should be able to tell by the different levels of WBCs in the blood. Platelets, okay, also called thrombocytes, are fragments, okay, of megakaryocytes, large nucleus cells developed there from hemopoietic stem cells in response to thrombopoietin, okay, which is secreted from the liver and the kidney. Platelets help repair damaged blood vessels by adhering to their broken edges, okay? So blood platelets, also called thrombocytes, okay, these are developed from hemopoietic stem cells in response to thrombopoietin. That's the hormone there responsible for them being made, all right? Secreted there by the liver and the kidney. These platelets there, these ones here, they help if there's a cut or any type of damaged tissue to repair it, okay? Normal platelet count there, anywhere from 150 to 350,000 platelets there per cubic millimeter. Megakaryocyte, okay, large bone marrow cell with a lobated nucleus responsible for the production of blood thrombocytes, platelets, okay. So megakaryocytes are involved in thrombocyte production, which are necessary for normal blood clotting. Megakaryocytes usually account for 1 out of every 10 bone marrow cells in normal people, but can increase in number to nearly tenfold during the course of certain diseases. So the number of these here can be increased depending upon what you have. Fun fact there, a platelet is not a true blood cell because it is only a fragment of a megakaryocyte. You remember that there from here. Okay. The blood platelets or thrombocytes are fragments of these megakaryocytes. Okay, good. Beautiful little slide here that just goes into all sorts of detail. I would pull from this and make little notes just so you have it. This is a beautiful slide. I should have put an asterisk, but I'm putting an asterisk there now. Have a look at that. Wonderful detail. Definitely a question or two from here. Plasma is the clear straw-colored fluid portion of the blood. Okay, plasma is mostly water. It's about 92%. Plasma constitutes about 55% of the blood itself and tends to have a pH in this range. And I mean in that range. It does not vary much. Okay, like I mean 7.15, you're now down into acidosis. 7.6, you're now into alkyldosis, okay, blood pH poisoning, all right. The plasma proteins are the most abundantly dissolved substances in the plasma, okay, which are not used for energy and fall into 3 groups. As mentioned there earlier, there's albumins, globulins, and fibrinogen, okay. The albumins help maintain the colloid osmotic pressure of the blood and account for about 60% of the plasma proteins. The globulins there comprise about 36% of the plasma proteins, designated alpha, beta, and gamma. Okay, alpha and beta globulins found in transporting lipids and fat-soluble vitamins. Gamma globulins, these are the type there that we use for antibodies. Very importante. Fibrinogen there plays a primary role in blood coagulation. Okay, kind of like a spider web, and it's able there to make that blood coagulate when needed, just in the case of a paper cut, right? Just more on the plasma proteins there. Love it. Origins of these guys there. There's your gamma globulins. These come from the lymphatic tissue. You'll see that these ones here all come there from the liver. Okay, these ones here are involved in antibody for immunity. Perfect. Gases and nutrients. The most important blood gases are oxygen and carbon dioxide. The plasma nutrients include amino acids, monosaccharides. These are single sugars, okay? Nucleotides from DNA, lipids, fats from the digestive tract. Since fats are not soluble in the water of the plasma, they are surrounded by protein molecules for transport through the bloodstream as lipoproteins. Fatty proteins. Good. Non-protein nitrogenous substances. Now, like the whole idea there, why, we say non-protein, because protein, and before I go into protein, the whole idea, the body will utilize carbohydrates first, sugars, right? That's easiest for the body to break down. And then if in a fasting situation, if there are no sugars, then the body will go after fats. And then further on, if there's no fats for the body to digest, it'll go on to and try and digest the body's leftover muscle. The problem with muscle, though, is that it has nitrogen. And this is trouble when you're talking about digestion. That you don't really get a lot of energy return on muscle. So here we're talking about non-protein nitrogenous substances. So these are substances there that are not protein based, but they have a high nitrogen content, because nitrogen is a problem, generally speaking. Non-protein nitrogenous substances generally include amino acids, urea, uric acid, creatine, and creatinine. Urea and uric acid are both the byproducts of protein and nucleic acid catabolization. Amino acids come from the breakdown of proteins, as we know. Creatinine comes from creatine, okay, creatine phosphate in muscles there, okay. Plasma electrolytes are absorbed by the intestine or are byproducts of cellular metabolism. They include sodium, potassium, calcium, magnesium, chloride, bicarbonate, phosphate, and sulfate ions. I didn't throw the chemistry in here, but you should be able to reproduce those. Bicarbonate is HCO3 minus, phosphate is PO4 minus 3. There's sulfate ions there. There's different ones, calcium. So just know your ions, and any trouble, ask me, and we'll get those to you lickety split. Some of these ions are important in maintaining osmotic pressure and pH of the plasma. Sure. Just a little recap here of where we have been. Okay. There's your formed elements. We've chatted about all those, all of these right there. There's all your there's your proteins. We're good to go. Hemostasis refers to the stoppage of bleeding. Okay, there's a great little link right there. Following injury to a vessel, 3 steps occur in hemostasis. Now, here we're talking about the whole idea when you get a cut. And when you cut and you bleed, you've cut a capillary. You've cut, and hopefully not an artery, but you've cut something, and that vessel now is cut in half, or you've sliced it somehow. You've opened it. It's broken. It's separate. One, which used to be one continuous pipe, has now been cut into 2 by something there that you've done. That's what we're talking about, the process here of blood coagulation, stopping that cut. So you'll see here the 3, you've got blood vessel spasm, absolutely necessary. The blood vessel itself starts to freak out and wave, and we'll show you. Platelet plug formation. You can kind of get an idea of what's going on there, and then eventually with blood coagulation. Let's go. So you'll see there that cutting a blood vessel causes the muscle in the wall, because you know that there's smooth muscle in there, right? Causes the smooth muscle in the wall to contract in a reflex or engage in what's called a vasospasm. You'll see what's going on here. This reflex lasts only a few minutes, but it lasts long enough, about 30 minutes there, to initiate the 2nd and 3rd steps of hemostasis. Platelets release serotonin. Okay, there's a neurotransmitter at this time, constricting smooth muscles in the blood vessel. Okay, so the release of serotonin, what it does is it tells the brain there that, look, we need these blood vessels to be constricted. And constricting the blood vessels does what to blood pressure? That's okay, I'm waiting. Constricting the blood vessels does what to blood pressure? You're squeezing them, you're making them smaller. So just think of Boyle's law, you're decreasing the volume, so you're increasing the pressure. Perfect. Okay, so that's what's going on here. And you'll see that what we call platelet plug formation. The platelets adhere to damaged lining of the blood vessel there within 1 to 5 seconds after injury. Sticky platelets secrete chemical signals, okay, and they control these microhemorrhages. Love that word, microhemorrhages. Hemorrhages is bleeding, right? may cause intermittent arterial microvascular occlusions and some peripheral vascular diseases. Okay, and you'll see what's going on here. That the break in the blood vessel, okay, and the blood escapes through the break, okay, blood is escaping, the platelets adhere to each other, they're really sticky, okay, and then once one there, another one comes, and you'll see what goes on here. The platelet plugs help to control the blood loss. Hemophilia, oh, I like these ones. I would give this one a read. Hemophilia is a genetic disorder characterized by deficiencies in clotting factors. Aha. Vitamin K deficiency, decreased synthesis of clotting factor needed to have healthy gut bacteria. That's all that healthy bacteria eat your yogurt. That's all in the large intestine. These bacteria, we give them a free place to live, and in return they give us vitamin K, which we need there for this whole rigmarole. Remember the whole idea of fibrinogen. That's what's going on here. You see those spider web there? It just holds all those RBCs in, right? The most effective hemostatic mechanism includes a series of biochemical reactions called clotting factors, which convert the plasma protein fibrinogen into the insoluble sticky threads of the protein fibrin. Fibrin sticks to the exposed surface of the blood vessels, creating a spider web which traps blood cells and platelets. The clear yellow liquid that remains after a blood clot is formed is called serum, which is the plasma minus the clotting factors. So, good stuff. Good stuff. Blood types based on cell markers or antigens. Now, you will remember those antigens. These are these glycoproteins there that stick outside of a cell that are white blood cells. They use to differentiate, okay, this is our cell as opposed to a foreign invader, right? Generally, we use an A, B, RH, which are the most important there when we're dealing with transfusions and newborn survival. Okay, agglutinins, antibodies dissolved in plasma that react with specific blood group antigens. Agglutinogens. Oh, this is what we're talking about here. Agglutinogens. If there is a foreign invader, these are the molecules there that are going to grab these foreign invaders and drag them down. So if you get somebody else's blood that you shouldn't be getting injected into you, what will happen is these agglutinogens will just grab this blood and just turn it into jam. So it doesn't flow, it doesn't go any further. Okay. Transfusion reaction prevented by blood typing and cross-matching for sure. Okay. Blood groups and transfusions. Have a look at this there. It's a nice little video. Pause the video, give it a watch. You know, we've got all these here. Just remember it's A, B, and then positive and negative has to do with your RH factor, which we'll get into there at the end. Okay. After mixed success with transfusion, scientists determined that blood was of different types and only certain combinations were compatible. Before a transfusion is given, the blood must be typed and cross-matched. The ABO system. Now there are many different other ways of differentiating one person's blood from another, but generally there we use the ABO-RH system. Okay. Groups named according to antigens present on the RBC membrane. Again, those glycoproteins that are sticking outside of the RBCs. Okay, there's type A, so there's antigen A, which is present on the RBCs. There is type B, where antigen B is present on the RBC membrane. Not A, only B. This guy here only has A type antigen. This one here only has B type. There is type AB, where it has both antigen A and B. And then there is type O. It has neither antigen. Doesn't have antigen A or antigen B. I'll show you. And we'll show you some slides here. This is what's going on here, right? So there's your type A, B, AB, and O, right? So type A has antigen A sticking out of these. Are those glycoproteins, right? Type B has type B antigens, a different kind. Type AB has both kinds, and type O has neither. Okay, just to show you that right here. Let's go back. Antigens and antibodies. Clumping of RBC cells following transfusion is called agglutination. That's what I was just taught. That's the jam. Okay. Agglutination is due to the interaction of proteins on the surface of red blood cells with certain antibodies carried in the plasma. So antibodies will react with these proteins on the cell surface. All right. We'll get more into that there again, but that's the whole process. If you dump somebody else's blood inside of you, these antibodies will recognize that, hey, this blood doesn't have the right antigens. It doesn't have the right glycoprotein, so therefore, by the process of elimination, it must be a foreign invader and it has to be taken down and turned into jam, agglutination. Only a few of the antigens on RBC cells produce transfusion reactions. These include the AB group and the RH group here. And just going back into the slide here, we've gone over the antigens for the 4 different types. But within the 4 different types, there is also the antibodies that are floating around in the plasma. Now, this type of blood right here, type A, it has type A antigen, but it has type B antibody. So it's looking for these guys. You see that? And as long as there's none of these guys in this person's blood, everything's hunky-dory. Everything's good. Okay? Type B, here we've got type B antigens, and we've got type A antibody. So you see these guys here, they're looking for these ones here. As long as we don't put this person's blood into here, this individual will be fine, because this is not going to react with these guys. This one here, you see its triangle, is not going to react with these guys here. But if you mix, this guy here will jam this one up. These guys here will jam this one up. Okay. The third type there is type AB. You'll see there it actually has both types of antigens, but it doesn't have any of the antibodies. Interesting. And then the universal, here we've got type O that doesn't have any antigens on it. So, but you'll see there it has types A and B. So if we could remove these and just inject this blood into here, into here, into here. Remember, you're not putting the serum in, you're just putting the RBCs in. So if you just put this blood in, you see that it doesn't have any antigens at all, so no reaction, no reaction, no reaction. Okay? And just another shot there, the same thing. If you didn't like the previous slide, well, my goodness, I hope you like this one. Okay? There's your RBC with your type A antigen. These are your antibody Bs. Okay? Here's your type B blood with B type antigen. And here we've got type A antibody. And AB, none, no antibodies, but both types of antigens. And you'll see there for type O, you have both types of antibodies, but no antigens there. So if we can get rid of these guys here and just inject the RBCs, it becomes now a universal donor. So who can give blood? Okay, so type A can receive from A or O. Type B can receive from B or O. AB can receive from A, B, AB, O. Okay. Why is that? No antibodies. None. Doesn't have, these antibodies here aren't going to attack this one. These antibodies here aren't going to attack because there isn't any, right? So, AB can receive from anybody. O, okay, can be given to anybody, but O can only receive O. Okay. Interesting stuff. RH, we're down to the last couple slides here. This one here is quite devastating and is 100% checked in all women, generally around the world if the medical facilities are available, and it has to be, because this one here can be devastating. Okay. There is another type of antigen, another type of blood system. There's other ones besides ABO, but this one here is RH. Okay. The RH blood group was named after the rhesus monkey and the test there that it was done on. If the RH factor surface protein, glycoprotein, is present on an RBC, the blood is RH positive. So if the protein is there on the RBC, the rhesus factor, they're positive. If it's absent, the blood is negative. Okay, if the protein is there on the cell membrane, Rh positive. If it's not there, it's Rh negative. There are no corresponding antibodies in the plasma unless a person with Rh negative blood has physical contact with an Rh positive blood. The person will then develop antibodies for the Rh factor. So, if you're Rh positive, you got the glycoproteins, and as well you have the antibodies floating in the blood. If you're Rh negative, you don't have them, and you don't have the antibodies. But you can develop the antibodies by having contact with an Rh positive person. There are two ways in which an Rh negative individual can have contact with an Rh positive blood: a transfusion or pregnancy. If an Rh negative woman carries an Rh positive baby, that would mean that dad, the father was Rh positive. So if an Rh negative woman carries an Rh positive baby, she may be exposed to the positive blood during delivery. The mother will now make antibodies against Rh that could attack the blood of a second Rh positive baby. And this is called erythroblastosis fetalis. And you see there, if mama is negative, and baby's positive, it's okay. Baby's going to grow up fine. Everything's fine. The delivery should be okay. But, there could be some blood upon the delivery, and the mom could be exposed to that Rh positive blood, which she would now start to develop an immunity to it. So now the problem isn't first baby, the problem is second baby. Now, if mama now has immunity, to this Rh, and if the second child is Rh positive, this can, mama's body will start to attack baby, erythroblastosis fetalis. Big problem. The problem can be prevented by giving the mother the drug Rogam. And you'll see what's going on here. Their mama is negative, baby is positive, but during childbirth there, some of the Rh positive fetus may enter the female's bloodstream. So now mama is developing immunity. Now mama is fully immune. And now if second baby there is positive, now we have trouble. So that is a good little introduction there to blood and its constituents. I hope you guys had fun. Go back and watch it. Definitely read the textbook as needed. And if you guys need anything there from me, let me know. But you guys take care, and we'll see you soon for cardiovascular. Bye-bye.